Hello, everyone. This is Mubena. Welcome you all in this virtual program. It is the 15th online discussion organized by Other Health Bangladesh. Our today's speaker is Peter Thomson from University of Sydney, Australia. And he will be talking on role of genomics for developing country agriculture. Before going to talk, let's know a little bit about Peter Thomson. Peter Thomson is an associate professor of biometry at the University of Sydney. His academic training was in geology and statistics. In his career, Peter was involved in both teaching and research. Peter has spent most of his career in teaching applied statistics to agricultural, veterinary, and medical audiences. His research background is quite broad too. Peter's main focus was on quantitative genetics and epidemiology to deal with big data. However, interestingly, his interests are in international agriculture development, assisting in country researchers for implementing and evaluating their programs, as well as developing their research skills. Peter has been involved in development projects and capacity building in Kenya, Zambia, Tanzania, Tibet, Vietnam, Pakistan, and Bangladesh. I would also like to take the opportunity to say that Peter Thomson is one of my PhD supervisors, and he is the best supervisor I have worked so far. I'm very excited, Peter, to have you here today. As a panelist, we have several distinguished guests present in this studio. We got Professor Ghulam Shahi Alam Sir, member, Bangladesh Accreditation Council, and ex-Vice Chancellor, Sylhet Agriculture University. Professor Alam retired from Department of Surgery and Obstetrics, Bangladesh Agriculture University in 2017 as a professor. There is also Professor Abdul Samad, ex-Dean and Director and Professor, Maharashtra Animal and Fisheries Sciences University, India. He is a dairy expert and spends lots of time in field level. Probably Professor Samad is connecting with us very soon. Here we also got Professor Dr. Samsul Alam Bhuiya, Department of Animal Breeding and Genetics, Bangladesh Agriculture University. We got Professor Dr. Ashutosh Das, Department of Genetics and Animal Breeding, Chittagong Veterinary and Animal Sciences University. There is Dr. Shofikur Rahman, Consultant, American Dairy. At the end of the session, Professor Ehsanul Haq will draw a conclusion. I also welcome all field-bearers and scientists, farmers, and students watching this program. Thank you all for being here in this afternoon. Now let's welcome Peter Thomson for his talk. Peter, the microphone is yours. You may start now.
Okay, thank you. Share my screen. I'm just trying to, Mona, just checking you've allowed me to enter.
Sure.
Delaware is, I think, Peter, so are you having problem to share the slide?
Just with sharing, yes. So let me just try it again. Oh, I think
Yes,
it might be share the screen.
Please take your time, yeah.
Yes.
You just click on the share button,
Yes.
just beside the cam or mic. Just click on the share button.
Yes, I've done that, and then I'll click on share screen.
Yeah, and click the share screen, right?
Yes, and share screen. Oh.
Sorry for the interruption. Maybe he is joining again.
দেলোয়ার হোস্ট কে তুমি নাকি?
না স্যার, হোস্ট হচ্ছে ম্যাম স্যার।
Okay.
হোস্ট
Fine.
তাকে কো-হোস্ট করে দিলে তাহলে আমার মনে হয় যে
Share screen. Entire screen. Okay. Now, can people see my screen now?
Yes, Peter.
Thank you. That is a relief. Okay, now let me just get started and right. Good. Okay. Sorry, a minor bit of panic. Well, first off, thank you very much for this invitation to present at this seminar. And first off, thank you, Dr. Mona, for that lovely introduction. And I will comment publicly, I thoroughly enjoyed supervising Mona as a PhD student. Also, I'd like to thank Dr. Mizzen from CBSAU for this invitation. Also, I hope this will be of interest to everyone for this Udder Health Bangladesh presentation. So I hope it'll cover a range of interests. So thank you for attending, especially on the weekend. I'm sure you've got many things to do, so I definitely appreciate the time that all of you have taken out to attend my talk. So just a bit of an outline of what I'll be talking about. I'll start out with a bit on the history of animal breeding and the role of genetics in animal breeding. But don't worry, we're not going to get into a high level of genetics. It really is the basic concepts. We'll then talk about the constraints of livestock improvement in developing countries. And then I guess the core part of the role of genomics in developing country animal agriculture. But I first need to talk about those first two sections. And I should say this is very much a personal perspective. If you ask someone else, they might have slightly different opinions. So I need to give you this warning. But I guess this is from my experience and observations of being working in this area for a while. So there's been a very long history of animal breeding, going back to the very first species that was domesticated. And that goes back to the dog that was domesticated about 15,000 years ago in Eurasia. And then in the table in front of you, you can see the approximate dates of domestication of many species. Sheep was about 11,000 years ago. And quite surprisingly, and you may well know it, that the duck is a fairly recent species to be domesticated, only 1,000 years ago. And what you'll notice actually is that there are many species that were domesticated in South Asia, Southwestern Asia, etc., Southeast Asia. So most species really were domesticated in your part of the world. So we've been doing animal breeding for a very, very long time. So let me just smooth this out. So I want to talk about various types of selection, how livestock and even crops for that matter have been improved. What you might call phenotype-based selection. And I suppose I should just add what I mean by phenotype. A phenotype is any observed trait or characteristic of an animal or a plant for that matter. So it could be a phenotype, could be the animal's coat color, it could be their milk production, it could be their temperament, any sort of observed characteristic. Now, from prehistoric times, livestock improvement or breed selection improvement has been done by selecting animals with favourable phenotypic characteristics. And these characteristics may be behavioural, and behaviour in the early days was very important. Breeders needed to remove some of the wild, maybe aggressive characteristics of animals. So behavioural, production, so they selected animals based on production, whether it be milk yield or fibre, and obviously reproduction, because you want to select animals that breed fairly quickly, and even appeal. Just the look of an animal has been very important, and this has been going on since very, very early days, as we saw up to 15,000 years ago with the dog. And the early farmers were able to progressively modify wild species to develop as agricultural species or companion species. And although this type of selection, of phenotypic selection, started out 15,000 years ago, it still is a very, very common form of selection. And this graph, this little picture down here, it just shows, for example, that selection for the development of the modern cattle, starting from the ancient aurochs, and progressively through selection, been able to modify the species to the kind of cattle species we see today. So let's dig a little bit deeper into what we mean by phenotype-based selection. So in other words, selection based on the observed characteristics of animals. And basically what we do is that we select animals to breed that have got favourable phenotypic characteristics. And from a genetic perspective, by that breeding, we increase the frequency of the favourable genes, or more specifically, favourable alleles in the population, and similarly, we reduce the frequency of unfavourable alleles. And progressively over time, we get the kind of livestock improvement that we're after. And this has been going on for thousands of years. But if we dig a little bit deeper, one of the simplest equations or models used by animal geneticists is the idea that the phenotype of an animal, or its observed characteristic, is a combination of the genetics and environment. If you like, P equals G plus E. Now, I'm a statistician, as Dr. Mona mentioned, so I can't help putting up equations, but this is as complex as it will be. And of course, this is a simplification of a model, but it's an important one in understanding principles of genetics. Now, phenotypic selection is basically based on P, just purely on the observed phenotype. We select animals with favourable characteristics, maybe biggest muscle depth or something like that. But it would be better if we could make selection based not so much on P, but more on G, because the G, the genetic, is what is transmitted from generation to generation. And the effect of E, the environment, and that's the management, which is still extremely important. Obviously, we need the best environment, we need to supply the best nutrition. But of course, that is not something that's transmitted across generations. So the effect of E is to add noise into the system. So, and if there's a lot of variation of phenotype due to non-genetic causes, then our rate of genetic improvement will be really quite slow. So that's the impact there. So it would be much nicer if we could actually select based on G, on the genetic part. So that leads to a second type of selection for breed improvement, what I might call pedigree-based selection. Now, breeders, animal breeders and plant breeders for that matter, have recognized that the phenotypes of relatives give additional clues to the value of an animal for breeding. They recognize that, a size, brothers or father, whatever, and similarly for a dam, its relatives if the relatives have got favourable phenotypes, then so might be some progeny. So they recognised for a long time that their relatives could give good clues about their value for breeding, and for a long time, this information has informally helped making breeding decisions. But it wasn't till the 20th century, the first half of the 20th century, with the development of quantitative genetic theory, that things really started to improve on this basis. People like Sir Ronald Fisher, J. Lush, etc., Henderson, they really started to make big improvements in quantitative genetic theory. And this genetic theory was utilised to develop what's called estimated breeding values. And these were developed in the 1960s by a geneticist, Charles Henderson. And basically, these estimated breeding values are the best estimates of that G part of the genetics. Now, in order to estimate breeding values, to estimate the genetic value of an animal in subsequent generations, it requires pedigree information, multi-generation pedigree information. So we need to know an animal's sire and dam, and its size, sire and dam, and its dam, sire and dam, etc. And going back as many generations as possible. And if we've got that pedigree information coupled with the phenotype information, then we can get a very good estimate of the G in that P equals G plus E. Phenotype equals genetics plus environment. So then we do our selection on G, not just P. So it's like removing the E, and we can do very much more rapid and accurate selection. So what we do, we would select animals with the best estimated breeding values, and we would breed from those, and that would lead to a much more rapid breed improvement. Now, just because that method was developed in the 60s, it took a while before it could be used, because there was it took some complex computation to derive these. At school, you might have solved linear equations, or basically, if you've got 20,000 animals, then there are 20,000 equations to solve. So you can get an idea that you need efficient computation to do this. And that really took till the 1970s before these were really start to put into use for making breeding decisions. So these EBVs, they've had a long use in Australia and other countries, but I guess I'm more familiar with the situation in Australia. So in Australia, EBVs, estimated breeding values, have been used for breeding decisions in dairy cattle. There was an organisation called the Australian Dairy Herd Improvement Scheme, now DataGene, that's been running since the 1970s. For beef cattle, a system called BreedPlan in the mid-1980s, sheep in about 1990, and then pigs 2001. And farmers and animal breeders will pay good dollars for EBV information. So it's not necessarily free. At least, some part might be free, but to get a lot of good information, farmers have to pay. But not only that, if a farmer then has a bull, a sire, which is with a very good EBV, that's now worth a lot of money to that farmer. So it's really become a very useful breeding tool, and also for farmers with elite bulls, it in itself has become a very good source of money. And these estimated breeding values, that's now been produced for a very wide range of traits, for production traits. So, for example, milk or protein content, etc. Fibre diameter for sheep. EBVs for temperament, because we would like to select for docile animals, even maybe to help to reduce horns in animals, health, EBVs are being developed for health. Now, and of course, these traits, these health traits, may well be other health traits. Now, of course, I know a number of people I'm speaking to here have got an interest in udder health because I'm talking to the Udder Health Bangladesh group. And in fact, one of my former PhD students, Mary Abdul Said, she's actually now working for DataGene, the group I just mentioned up here, and her role is to develop breeding values for udder health traits. So if anyone is interested, I can link you up with Mary and the DataGene group. So they're really now being produced for a wide number of traits. So that was phenotypic selection and then pedigree-based selection using EBVs. There was another form of selection that has been looked at, and its origins go back to the 1980s and 90s. This is called marker-assisted selection, or MAS. And in the 1980s and 90s, genetic markers were developed. And these are markers that can be used to determine the genotype, so the actual genetic type at a known position on a chromosome. Now, in the early days, these were very low marker densities, maybe 50 over the genome. So you might have, maybe if you're lucky, 3 or 4 at the most markers per chromosome. And what they allowed, though, was to look at the association between one of these markers on the genome, on a chromosome, at a known position, and the trait. So that allowed or spurred the development of what was called quantitative trait locus, or QTL mapping. And the idea of QTL mapping was to locate the region on the genome or of a specific chromosome that might affect a quantitative trait. I say quantitative in brackets because it can actually be used for any trait, really, not just quantitative. But it's known as quantitative trait locus or QTL mapping. But one of the troubles with QTL mapping is that it did find associations, but they tended to be very broad. And these very broad regions would possibly include many functional genes. So QTL mapping didn't determine individual genes that control, for example, milk protein content or stature, height of animals, etc. But just to show you some examples of QTL mapping, these are some QTL mapping exercises that I was involved in over my career in genetics. The top example is an example just from one chromosome, chromosome 1, of about 20 odd, 25, I think. I'm not sure how many chromosomes in the mouse. And what these show here, markers. So we have 1, 2, 3, 4, 5, 6 markers on the chromosome. And the trait being looked at, I haven't labeled it here, but it was a heart defect. It was a gap in the heart valve. So I was actually working with some hematologists. And the peak here represents a likely gene affecting these heart defects. So sure, the most likely position of this gene is up here, but it's a fairly broad peak. In fact, anything above this dashed line has got a significant association with the phenotype. So it's not a very sharp definition of an association. In the graph below, this is some work I've done with some colleagues working on sheep, and the trait or the phenotype being assessed here was the weight of sheep at 43 weeks of age. And this is a genome-wide study, so we've got every chromosome up to about 26. So I think there are 26 chromosomes in sheep. And again, the peaks indicate significant QTL. In other words, there are genes with a significant association of this body weight trait. And we can see a few peaks. The solid line is the confirmed line, if you like. The dash is suggestive. But you can see on sheep chromosome 6, there's a big peak here, indicating we have associations. So there are genes there. There's at least one gene there associated with weight, but it's actually still quite a wide region. It's about the third of the length or a quarter of the length of the chromosome. So, you know, it's not terribly sharp, but, you know, it certainly does indicate there are associations there on chromosome 6 and again down the end of chromosome 23. So that's an example or two examples of QTL mapping to try and find specific genes associated with quantitative traits. But the way it works, so once we've identified these peaks, the way that it was then supposed to work, how it linked up to marker-assisted selection or MAS, after we identified the QTL regions, we would then look at other animals and identify animals with this desirable, with the desirable marker genotypes, because the QTL mapping would tell you what markers to look at, and in particular which alleles are favourable, and then we would scan animals with those and find animals with at that particular region or regions with the desirable marker genotypes. And then we would select animals based on those for breeding. And the idea is that hopefully that would lead to genetic improvement, which sounded quite reasonable. And that was the plan in the 80s and 90s. However, unfortunately, we didn't get that gain that was hopeful. And there are a couple of reasons. One, I guess I've already mentioned, the broad location of QTL. These are not sharp regions. And the other issue too is that just because you've mapped a QTL in one population, if you then take it to another population, it may not be there. So suddenly the QTL has vanished. So unfortunately, the promise of QTL mapping linked to marker-assisted selection didn't always work. It did sometimes, but it wasn't this great leap forward that a lot of us who were working in quantitative genetics at the time were hoping for. So meantime, while all this was going on, pedigree-based selection using EBVs continued. But things started to change in this century, in the 21st century, where genotyping started to come in not by using old satellite markers, etc., but by using SNP chips. And the SNP, without, you know, for those who haven't done genetics, that doesn't really matter. A SNP is what's called a single nucleotide polymorphism, but it's variation at one of those base pairs. I mean, everyone probably knows that all the genetic sequence is made up of these 4 base pairs, AG, TC. And we're looking at, and the SNP gets down, is a marker at that very specific SNP location. And usually, or at least once the SNP chip is developed, we will know the specific location of that SNP on the genome. Now, the earliest SNP chips had 50,000 SNPs, so not just a handful like in QTL mapping, which we talked about 50 markers. Now we're talking about at an entry point of 50,000. So once SNP chips got developed, we had 1,000 times more markers, these SNP markers. And so the SNP chip that was made available then was called the bovine SNP50 chip. And since then, the density has increased, and they are now making available these bovine HD or high density SNPs with 777,000 SNPs. So very high density. So these SNPs are very tight together, unlike back here, this very low density microsatellite markers. So very, very, very dense. Now, I should say though that these SNP chips are available for cattle, of course, and the major agricultural species like sheep, goat, pig, chicken, salmon, and of course many plant agricultural species. And more SNP chips are being developed, but I think it's probably a bad term. I put they're minor species because they're not minor in many countries, are not covered by a SNP chip. So, for example, camels, crocodiles, I've worked on both of those species. I know you don't have too many camels in Bangladesh, but I do believe you have crocodiles. But these species are not covered by a SNP chip. But it is possible to do genotyping, what's called this GBS, or genotyping by sequencing method. And that allows genotyping of SNPs, but without the requirement of a chip. Because I think for some species, it'll be a long, long time if ever a chip is developed, because they're very expensive to develop a chip. But with GBS, the prices are now coming down, and it's maybe now become viable. Well, I believe it is viable to do these on other species like crocodiles, camels, other species that are not the major agricultural species. So then this allows another type of selection, genomic-based selection, which I'm now going to outline. So just to start off, we talked about pedigree. We talked about phenotype-based selection, and then we talked about pedigree-based, and then we talked about Markov-Sitz's selection, which had problems, and now onto genomic-based selection using these very high-density SNPs. Now, with that, we can get actually much more accurate breeding value estimates, what are called genomic EBVs, and and there are a lot of studies, both empirical and theoretical, that show you can have better selection potential using these genomic EBVs, where we brought in genomic information rather than just pedigree information. So, and using SNPs, we can conduct what are called Genome-Wide Association Studies, or GWAS, which you might think of being a very refined form of QTL mapping for finding SNP trait associations. Remember, QTL mapping gave you these very broad regions. Using SNPs coupled with GWAS, we can do a very refined form of mapping, or GWAS. So this is an example of a GWAS here. Now let me just explain. The trait being looked at here was the protein percent in milk of Australian dairy cattle. And this research actually was conducted by Santi Pangmao, who did a PhD with us, and he's since returned to Thailand to start to work on some of these methods here, back in Thailand rather. But the idea of GWAS mapping, it's like QTL, but very high density. Every one of these dots is a SNP, and the height of the SNP indicates the strength of association. So what we're looking for is anything above, or particularly above, the red threshold line, which is the highly significant line. And what we can see quite a few areas, but particularly here on chromosome 14, so cattle chromosome 14, two very distinct, very sharp regions, and that may be more than one gene, but it will only just be a handful of functional genes within each of those regions. So it then becomes quite viable to then work out the actual functional gene involved. So it won't be hundreds of genes in those spike areas, maybe just 2 or 3 or 4, and they can be then followed up by other means. So you can see it's like QTL mapping, but it gives you very, very, very sharp peaks, not these broad peaks that we saw in QTL mapping using microsatellite markers. So, okay, so what we got up to this so far is kind of a range of techniques of animal breeding and some of these new genomic techniques that can be used for genomic breeding values or for GWAS to identify particular functional genes. So now what I'd like to do is talk a little bit about constraints to livestock improvement in developing countries. Now, again, this is personal observations, and probably you people could speak to this far more than I can because you're right in the thick of it. But as an observer, this is kind of my thoughts on it. And I guess the main thing is we cannot just simply import elite genetics from developed countries for livestock improvement, because often maybe these elite Friesians, Holstein Friesians, might be optimized for very good nutritional plains, good, maybe a difference of environment, because we need to be aware of G by E, genetics by environment interaction. So just because a Friesian, a Holstein Friesian, might be great in the Netherlands, it may not be suitable in Bangladesh or Tibet. And for example, I actually did see one time in Tibet some Holstein Friesians that had been introduced, and they were in a far worse state than the local breed. So what we should be doing, I think, is utilizing locally adapted breeds that tolerate the environment and nutritional plane. And crossbreeds may also be useful. So, but to unpack this further, I think we need to consider two types of farms in developing countries. One is the medium and large farming enterprises, and then the smallholder. I guess my main interest and emphasis has been in smallholder farms. I've done work with this particularly in Pakistan and East Africa. But I think a lot of you maybe perhaps work on medium and larger farms. So I will make some comments there to start off with. So I guess in the medium and larger farming enterprises in developing countries, they are relatively few, at least compared to smallholders. But I think there's certainly a very substantial increase in these medium and larger farming enterprises in developing countries. I believe that's the case in Bangladesh. I've seen them in Pakistan and East Africa, various other places. And it's because they are addressing some consumer demand for dairy products, particularly in the larger urban areas. Now, although I mentioned the problems of importing elite genetics, it can work if it's coupled with some good husbandry, good nutrition, all those sorts of things. So, if that's done, I think, these medium and large ones can make some quite good progress. My observations, and I may be wrong, is there is limited use made of estimated breeding values for selecting breeding animals. They are being used in some places, but fairly limited, I believe. But so in terms of data collection on some of these systems, I think there's some very good in terms of collecting production data and health data, but often limited in amount of this pedigree depth going back, say, 5 generations. So this is where I think there might be a shortfall and something that I think should be addressed. So I would suggest in these medium to larger farming enterprises, pedigree recording, I think should be a priority to allow these development of EBV-based selection. The tools that are being used in, like in Australia that I mentioned, and other countries. But okay, what about SNP chips? So what's the capability there? Well, I think with the first SNP chips that came out, I don't think that the costs were very high. And I think originally the costs were too high to be useful in developing countries. Whereas more recently, cost of some of these SNP chips has declined considerably. And I think for some of the medium, particularly the large enterprises, it is worth now considering, at least in the future anyway, use of some of these SNP methods. And then we could use some genomic-based selection, then might be viable. So if you forgive me to make some recommendations, and as I say, a lot of what I'm talking about is my own personal interpretation views. I guess for these medium and large enterprises, I'd suggest to start, well, obviously get pedigree information and then start with EBVs using pedigree-based selection, and then progress to genomic-based selection. I would also be suggesting to try and set up enterprises across the nation, a nationwide one. In fact, if you could go region-wide one, even better, because a lot of the accuracy in EBVs comes from very large databases. And so rather than every sort of, enterprise having their own, if there's some way to link across enterprises, maybe sponsored by government, veterinary institutes or something, I'm not sure. But what about smallholders, smallholder farms in developing countries? And I guess I probably spent more time thinking about this area and working in this area. So across the world, there are something like 570 million farms, and 90% of these are smallholder, according to the FAO. And they're very often, these smallholder farms, there's a lot of poverty, or poor, they're poor anyway, if not poverty. There's food insecurity, and they're very vulnerable to climate change. And of course, climate change, I know, is a very important issue there in Bangladesh. I know there's a lot of thought about issues there. Another thing to keep in mind for smallholder farms is that they often keep lots of local breeds. And so I tend to think of smallholder farmers as being, they maintain genetic diversity. They're keepers of genetic diversity, which is a great resource for the future. I just found a reference earlier today that 85% of cattle are indigenous in Bangladesh. I'm sure you know all about that. And here are just some of the breeds which, well, in fact, I'm sure you know all about that too. I won't pretend to know much about that. But anyway, I found it very interesting that 85% of cattle there are indigenous. So there's a lot to conserve in terms of that local genetic diversity. And I think it's this amazing resource. We need to maintain that diversity as a future proof against climate change, because we don't want a situation where the world is dominated by Holstein Friesians that's only will be working in one environment. We really need to maintain that diversity into the future. So and as I say, it's often smallholder farmers who maintain that diversity, and we really need to see what diversity is out there. Excuse me. So, in terms of how improvements can be made for smallholder farms, well, I think obviously extension training for things like nutrition and husbandry. I would argue a whole family approach, men, women, and children, is important for development, and also a One Health perspective, because in rural areas there is an intimate association between people and the livestock they own. But I think breed improvement, selection has got an important role to improve livestock there. Currently, I think breeding is in smallholder is done a bit opportunistically in terms of it's limited by the farmer's choice. What animals they have, in either their own or their friends to breed from. So what type of selection can we use? Phenotype-based selection? Yes, that's happening. Maybe not as thorough as we might like, but yes, people do select animals based on their favourable characteristics. What about pedigree-based selection in smallholder? Well, I will say no, because it's a bit not reasonable to ask smallholder farmers to keep record of pedigree. Sometimes they may not even know which animals mated. So I don't think it's viable, and I don't think we can ask smallholders to necessarily keep those records. Marker-assisted selection, I think I already eliminated that before. We decided, that it wasn't particularly useful. Whereas I believe genomic-based selection does have a role. If you asked me 5 years ago, I would have said no, but now I think it does for some reasons I'm going to explain. So one of the things to make improvements, EBVs are a great tool, but we can't, and for that we need pedigree. But we can get around pedigree we can infer pedigree from genomics. If we have, SNPs, and we don't need that many SNPs, we can actually infer relatedness. We can work out, with a certain degree of error, of course, an animal's sire and dam and their sire and dam, etc., from the SNP chips. So, and we only need a relatively small number of SNPs. And what are being produced now are some customized low-density chips. So they could be utilized to infer pedigree. So we don't need to ask a smallholder farmer to record it. And as I said, genotyping by sequencing costs are coming down. That will be an alternative to species that don't have SNP chips. And then we can generate EBVs or genomic EBVs using this, where we've inferred the pedigree from the SNPs. But of course, there's a lot more to consider for breed improvement other than computing EBVs. There are many, many logistical aspects that need to be considered. So what's the way forward for smallholders? So we need to consider, if we're going to do this, a lot of logistics need to be considered. We need to consider who and how the phenotypic data would be collected. Milk production, weight of an animal, or girth of an animal. So they need to be collected and taken to some centre, a government department, university, for processing. And then who does the data processing? And more importantly, how is that information going to be sent back for farmers for their breeding decisions? So it has to get back to them. And maybe we need to work on getting support from a focal farmer, an important farmer in a village. Maybe we work with focal farmers and they then provide superior breeding size for the whole village. So these are some of the logistics that might need to be worked out. Importantly, we need involvement of farmers for the decision-making process. We can't come in, even a government department can't come in and impose. I really believe we need the involvement in the farmers for the decision-making process. And for that, we need to know where farmers get their information from, who they get it from. And from some other studies I've been involved in, they don't necessarily get them from their district veterinary office. So we really need to work out where people get their information from and work with that. So just to finish off, I believe that genomics does have a role in developing country agriculture, but we need to record data. That really is key. Without data, we can't make good breeding decisions. So there are a lot of logistic difficulties, but we really have to do it. We have to get stuck into it, I believe, because we need to do something to help particularly smallholder farmers, because where they are living, they are being subjected to a lot of environmental change, environmental pressures, population pressures, coupled with climate change. So big issues that I think need to be solved to address their life and food supply, etc. And as I say, I'm certainly not the only one who's thinking about these issues. There's a group primarily based in Africa, Nairobi, the International Livestock Research Institute, but they also do work globally. I believe they have an office or a campus in India, and they really support livestock research and livestock improvement around the world in developing countries. So they could be a group that you might want to make some contact with. Just to finish off, I'm showing some photos. I apologize, they're not from Bangladesh. You might guess they're from Pakistan, where I've done some work. The reason I'm showing this photo is to emphasize people need to be involved. This is all a photo about people. So on the top left, working with the men of the village. Remember I said it's very much a whole family approach. So this is working with the men of the village. And then over further to the right, we've got working with the women in the village. The very back, actually, you might see me and a few others. And then below, straight underneath, we've got a training session. In fact, one of those women there, she did a PhD in Australia recently. And then right over on the right, some school children. As I say, it's a whole family approach. I went to this school and these school kids were describing how to diagnose mastitis. Fantastic. And then right over at the bottom left, these were a collection of farmers who had medium to large enterprises, and they are talking about some breed management decisions. So really getting everyone involved in this, it's really important. We can't just impose from the outside. Anyway, I'll finish there, and I'd be very happy to take any questions.
So this is time for question and comments from the panelists. I would like to request Professor Ghulam Shahi sir, please. Sir, you might need to
please unmute the mic.
Sorry. Thank you, Peter, for your nice talk on breeding policy and policies what we could adopt for the development of our livestock population in this country. We have started crossbreeding from a long time ago, and we are using the Holstein Friesian cow to upgrade our local stock. And the situation nowadays, the first service concession rate is going down up to 27% nowadays, and the calving percentage also very reduces up to 40%. So you have just rightly pointed out, the country like us, we need to very carefully select our animal on the basis of genetic nomenclature or genetic based the genomic based selection or nucleotide polymorphism, etc. But the country, for the genomic approach for animal production in developing countries, okay. For us, it's very challenging, no doubt of it. We need to have, you have rightly pointed out, we need to have a good recording system that we are lacking on. So thank you, Peter. You have rightly pointed out the situation that we are facing at this time. We need to develop our breeding system. Policymakers should take the decision rightly. Otherwise, it would be a catastrophic for us in the next or the future generation. Thank you, Peter. Thank
Yeah,
you.
look, thank you. So, yeah, look, there certainly will be a lot of logistic decisions. And as I say, I think different management strategies will be needed for smallholders versus the medium and large holders. I think the real challenge will be for smallholders, but we have to do something to help them, I believe. Yeah.
You have to be very careful in selecting animal and what sorts of breed we need to use for developing the stock. It should be carefully should be done. Otherwise, that would be will be end up with nothing.
Yes. Look, I would agree. I would agree.
You know the situation of Holstein Friesian all over the world nowadays. So I think this is the time the policymakers should come forward and consider these things for developing policy of livestock development or dairy development or anything like that. Yes, Peter, thank you.
Thank you.
Thank you, Peter. Now I would like to request Professor Abdul Samad sir for his question and comments. Welcome, sir.
Thank you. Thank you very much. Hi to everyone. Really happy to be, listening to your lecture, Peter. I'm from Mumbai, India. Work with a lot of smallhold farmers, and have also been involved in spreading data recording system. Specifically for helping the farmers manage their farms more efficiently. And the other thing we are doing is also evaluating sire in terms of fertility to advise the farmers which sire to be used in a particular area, because you have rightly pointed out that a genetically superior sire semen imported from abroad may not be of the same ranking in the tropical climate. And similarly, we find that if there are 50 sires, sire A might be good in an area X, but is not in area Y. So our system keeps an eye on conception rates sire wise, so we can also advise it. Well, coming back to today's topic, I had a couple of questions for my own knowledge enrichment. The one is you said that and you have said very rightly that probably genomic selection and going through all those all that SNP and others might be very a very long drawn process, very expensive process for developing countries. Can we not have a program where we import say genomically proven sires sex semen, and through IVF produce enough of heifers and do progeny testing also? I mean, the classical progeny testing to narrow down the selection process. And fortunately, there are also good like Indian breeds which are suitable for tropical climate, which have been genomically proven, say in Brazil, and get their sex semen and do a conventional progeny testing. So is that a possibility? Has anybody tried that? That is my question number one. And if you may allow another question, when we talk of crossbreeding, it's basically a stabilization and producing a synthetic breed. Going haphazard way, unless and until after F1, you have got enough of indigenous, and then you are crossing them, and also having indigenous breed, and your terminal cross goes for meat industry. Then it is fine. Otherwise, we will have to have a stabilization process and come out with a breed which is stable breed, like Girolando, which has been done in Brazil. But that kind of activity is possible only if you have good recording system, and then you can do crossing and back crossing to come to that 3/5, 3/8 and 5/8 status. That is probably very, very, very difficult. So is there any shortcut for say taking the existing breed of crossbreds and reverse crossing them to bring them to stability? So these are two questions from my side, and I think we have been giving a lot of emphasis on selection, and I was talking to our Bangladesh colleagues. There also seem to be issue with culling of animals, and are there criteria for culling of animals? Say for example, instead of having a cow maintaining it for say 4 or 5 years, and then find out that it is not giving enough of milk and it goes out of the herd, can I select these animals say at the stage of 6 months or 9 months based on some phenotypic principles? So these are my questions which I would like Peter to answer. Thank you.
Okay, well, there were 3 questions.
That's right.
So I'll try and answer the third one first while it's still in my memory. Yeah, that's a hard one. So I think you want to make a decision very early on if you want to cull. Well, I guess breeding values will tell you that, because what you're trying to do is make some indication about something in the future that you haven't actually measured yet. Well, that to me is the value of a breeding value. It tells you, I mean, you can have a breeding value of an animal even before you got any phenotypic information. So that might be, partly part of what you've done. But the other thing too is that I'm a statistician. I do a lot of modelling, and there may be some information, some signal in the early milk production that will make future predictions. So I will say I'm probably more of a statistician than a geneticist. So that's the path I would see. Can I do that sort of forward prediction? But I think the EBVs will tell you, because if the age at which the milk production goes down is a heritable trait, then that would be amenable to developing an EBV for the rate at which the milk declines with age. So it could be selected upon.
Actually, Peter, my idea was, since milk production or milk synthesis is a biochemical process. Okay, so a cow which is giving say 30 liters of milk would be having enzymes, whose turnover rate must be higher than enzymes, from a cow which is giving say 5 liters of milk.
Yeah.
So are there such on a biochemical test, which can be done on the tissues of the cow, heifer, because even if it is not giving milk, those genes might be there, which if it is stimulated, would be able to tell us whether a particular cow is having an enzyme which is having right turnover rate.
Look, that sounds reasonable, but that I will have to say it's beyond my expertise. I don't think I can comment meaningfully on that one. Now, sorry, I have a bad memory, and I had meant to write it down. What was your first question?
The first question was, can we not import sex semen?
Oh, that's right. Yes.
Yeah.
Look, I think there's no reason why not, except though if you import it, you've got to make sure that you're importing genetics that will suit the environment. We always need to be aware of G by E. Now, if it's possible to import semen from an environment you think might be similar, then that's fine. But it's just always, I think it just seems a bit risky because they may not perform that well. I'm not sure that's answered your question.
No, what I was suggesting was, say for example, from Brazil, I import sex semen of 50 sires.
Yeah. Yeah.
and test in Bangladesh with sex semen and IVF, produce 20 heifers for each sire,
Yeah.
and
Yeah.
then within a period of 3 to 4 years, check, out of those 50, which are the one which are performing better in Bangladesh,
Yeah.
and then select them for insemination. So, is that a right approach?
I mean, I think that sounds very sensible. Yeah. Yeah. Yeah.
Okay, thank you, Peter and Professor Samad. Probably we can continue this discussion in our next project. That will
Thank you.
be.
Thank you, Peter.
Thank you.
Now I would like to request Professor Samsul Alam Bhuiya sir for his question and comments. Sir, please.
Okay. Thank you, Dr. Momena, and thank you, organizer, for nominating me as a panelist in this webinar. And I'd like to give sincere appreciation to Professor Peter for his informative and comprehensive deliberation. Particularly, this is my field, what I have been done for the last 2 decades. So, Professor Peter rightly mentioned about the genotype environment interaction, pheno-based selection, then pedigree-based selection, and finally the genome-based selection. And he rightly pointed out the record-keeping system is we cannot develop since we are practicing crossbreeding for the last 6 decades. We started our crossbreeding in 1962. Even though after 6 decades we cannot develop our record-keeping system, and the genome alone cannot walk through without the phenotype. So I have some queries to Professor Peter, not queries about the genomic selection. If we use only genome-based selection, 50K or 777K, and based on these, we calculate genomic relationship matrix, right?
Mm-hmm.
So what is the reliability for these genome-based selection, only genome-based selection? I mean the genome information guided selection. What is the reliability of the genomic prediction?
Yeah.
Is it 70% or 80% or it is? I have a little experience with Cedric Gondro,
Yes, I know. Yeah.
Yeah, so we found a relationship when we incorporate phenodata with pedigree as well as with the genotype together, then the accuracy goes higher. So another query is we are developing country, there is no recording system. So what is the optimum proportion of optimum population size for genomic prediction? So I have experience with South Korea. If it is more than 2,000 individuals, then the accuracy is relatively stable. Otherwise, the accuracy fluctuates too much. So, what is your experience, particularly in the East Africa, as well as in Australia? What are the minimum size for genomic prediction? Second one. And third one is why did you just discourage the pedigree-based selection? So far I know pedigree-based selection, the most of the breed has developed in the world based on the pedigree-based selection. So even though we have lots of limitation, particularly for a developing country, so can we just make a simplified pedigree record? Can we keep so can you suggest us for a simplified recording system developed for the pedigree, and then we can evaluate our animals? And you have to remember that we are we are practicing crossbreeding, but we are importing semen from Australia, Canada, New Zealand, like this. So importing the elite animal and just breed the native resource. So this is not the actual way. We have to develop our crossbred bull here in Bangladesh. Otherwise, we cannot develop our synthesis breed. So all together, I have 3 queries to you. So I would like to know. Thank you, Peter.
Okay. All good questions. Let me try and answer these.
Yeah.
So in terms of looking at relationships, so you're right. If you can use both, if you can develop a relationship matrix based on both the pedigree and the genomics, that's going to be better.
Yeah.
However, if you just compare say one based on the pedigree versus one based on genomics, the one based on genomics will be better in terms of if you're going to do selection. It will be more accurate if you base it on the genomics, on the SNPs, rather than purely on the pedigree. Of course, if you can have both the pedigree and the genomics, then you will do better. So, and I'm sure I know Cedric Gondreau, and I'm sure he could give you a better answer on that one. But basically, there's a lot of simulation work and empirical work that would say that if you use SNPs, it will give you a better result just than pedigree. Okay. The second question, how many do you need? I can't really give you a good answer to that, because most populations I'll be dealing with, I've had at least 2,000 records anyway. But I believe that if the numbers are very small, genomics may not be that good, then you might need a pedigree. But look,
that is the main barrier for our developing country. We are dealing with the small sample size, and which might not be fit with the in the prediction equation. We cannot fit it. This is the problem, and accuracy goes down when we use only limited number of samples.
Yes.
So first, you rightly mentioned, we have to develop data recording system at first.
That is critical.
Farmers, and another important issue, we have no breed society or farmer organization. Recently, we developed some farmer organization, but they are not they are not just exchange their data material to one another, just keep their data by himself or herself. So we have to accumulate the pheno-based data at first, then we can apply for the genomic genome-wide expression studies or genomic prediction for selection the superior bull or heifer. That is my so far I know. So that is my realization, and you can add up with it more or disagree with me.
Yeah, look, that's why one of the things I mentioned earlier is, at least for the medium and larger ones, to try and link up, if it were possible, to have a nationwide scheme, because that's where you really get the gains by increasing the size. So every little enterprise having their own will not be very powerful. So, and maybe it has to start out by starting with a breed society to try and encourage that. But, setting up something across the nation would be very, very useful. The third question, you said, why am I saying don't worry about pedigree? I was saying that in the context of smallholder. And the reason I'm saying that is that it will be very difficult to get smallholder farmers, at least my impression. It's going to be very hard to get smallholder farmers to record that pedigree information. And as I say, sometimes they may not even know. So that was my reason there of saying, we not to worry about the pedigree information for smallholders. For medium to large farms, then yes, we need pedigrees, absolutely, because I think it's easier to record pedigree information, I think, in a medium to large enterprise. But smallholders, I think it's going to be difficult. But that's just a personal opinion.
Yeah, you're right. So thank you so much, Peter, for your really excellent presentation with information, particularly for animal screening and selection. Thank you so much.
Thank you.
Thank you. This is time to hear question and comments from Professor Dash. Sir, please.
Thank you, Momena. I'm really feeling privileged to be here with all the knowledgeable scientists from different parts of the world. Thank you so much, Peter, for your excellent presentation. You correctly pointed all the features of selections, and you do recommended some of your personal perspective about the genomic selection or other things, what we can do here in developing countries. Lot many things was in my mind, but my previous discussion there discusses about those questions. I have some technicalities, a question about technicalities. You mentioned about low density SNP chip about the smallholder farmings, particularly the small size. Is it possible to make that kind of low density SNP with the higher genetic diversity? As you know, we don't rearing like a very specific breed. We have like a genetic admixture from different breeds and other things. This is the question number one. And is it possible while doing if we can do that, while doing the data analysis, can we adopt a new prediction equation considering those genetic diversity and agroclimatic heterogeneity? This is the question number two. And the question number three, normally for the data analysis, we know that it is difficult to choose the proper genetic evolution model. Despite of having this heterogeneity, do you suggest any particular genetic evolution model for our developing countries? So these three are my questions. Yeah.
So, sure. I mean, I don't see genetic diversity necessarily being an issue. I don't think it would prevent the application of a SNP chip from my perspective.
So in that sense, do you need a reference population? What do you think? Like if we go for the genomic selection?
I guess you're right. I haven't really thought through that one. That is the standard thing that's done. But look, from my perspective, I mean, in terms of calculating EBV, you don't need a reference population for that. I mean, all you really need, from my perspective, is just you've got a population there. You've done the genotyping. From those, you can get a relationship matrix, and then from that you can derive EBVs. That's my perspective. That's the kind of thing that I've done for my work here.
Okay. Yeah. Thank you. And my second question was about the development of prediction equation. Is it possible to develop a new prediction equation?
Well, yeah. I would think just a very the standard sort of mixed model approach, and that can be extended for multiple breeds if need be. I think just a standard mixed model, standard BLUP approach, I think would be fine. I don't see you need anything different. Yeah. I mean, for example, I've done some work like this in a small camel population, just to type the technique, and we were getting reasonable estimates of breeding values, and the whole thing was based on SNP data, and it seemed to be promising.
Yeah. Thank you so much, Peter.
That's fine.
Thank you, Dr. Dash, too. In the panelist, we also got Malik Omar. So I'll request Malik Omar if he has any question or comments in Peter's presentation.
Renowned dairy farmers.
Thank you very much. Actually, here everybody is academician, and I'm not from any institute. I am a dairy farmer, and I'm from the dairy farmers community. In our country, genomics is great problem, because if you go for genomics, you need all the phenotypic or pedigree data or genomic data of the bull. But practically in our country, our farmers got semen from the bull of different breeding stations, government or private. They don't even get the pedigree or father, mother, sire them information of bulls also. They only get a Frisian percentage. In our country, everybody gets semen, just one number or name is written, and then it's written only the Frisian 87%, Frisian 50%. No one is actually able to disclose, or they're able to give their pedigree information of the bulls also. So here, how genomics will be done? And my next question is, because if we are not getting any information, how it's possible to develop their flock? What is your suggestion for the farmers?
Okay, so the first point, I totally agree. It's going to be very difficult to get farmers to record the pedigree, and that's the beauty of genomics, is that the genomics will tell us, what's the sire, what's the dam, etc. See, that's the whole point. So, ideally, yes, we would have good pedigree records, but if we don't, then genomics can come to the rescue. That's my point. So I don't know if that addresses.
How genomics will help if there is no record of the bull also, rather than only Frisian percentage?
Well, okay, there has to be some record. Someone has to go out to the bull and get some information of the bull. They need to go out and get a hair sample or a blood sample or something like that. So you need some information. Yeah.
Now, if the information is not available, then what to do? Then how it's possible to select the bull?
You're going to need some information. I mean, the first thing you got to do is to get.
How will you evaluate? Without data, you cannot evaluate. Sorry.
Well, no. I mean, you need data. You cannot make decisions without data. That's the whole point. And so that's why I said it's not about the technical aspects of calculating a breeding value or anything like that. The first big stage is to get farmers on board, that they have to see the value of it, what it will return to them. So the technical aspects, the genetics, people have solved those issues, but I think the big challenge is getting farmers on board. That they have to see what the benefit would be, and maybe someone needs to do a cost-benefit analysis, some economist or something, and then take that information to farmers, to a village, and show this is the benefit you might expect. But you've got to get them on board, and the farmers have to be involved in the decision making. So it's not just us, me at a university going and saying this is what you do. It's got to be done. It's really got to be driven very much by the local farmers. They need to be on board. And if we don't get information, then no, we need that information.
Thank you. in our country, another problem in our farmers community as well as our consultants also, that the increase of Frisian blood percentage will solve everything. But practically, we are me and some other old long-time farmers, they found that the increase of Frisian blood percentage raise hazards in our tropical country. And you are from Australia. There are some tropical areas in your country also. But our common phenomena is that just raise of Holstein blood is everything. But it's actually it is getting hazard for our farmers, and they are losing their production. Some farms are being closed for increasing Holstein blood because it gives hazard. Then how to rescue our farmers and actually maintain and increase our production?
Well, I bring back local breeds. I probably would reverse bringing in too much Holstein Friesian. At least be very careful about that. I mean, I tend to think of a Holstein Friesian being like a high performance car. So, it's great if they're on a good road and they can be maintained quite well. But, if you suddenly put this high performance car on this really rough road, they're not going to go well. You need a four-wheel drive, you need a Land Rover. And I think it's like that for Friesians, that they're wonderful high performance cows, but they require a lot of nutrition to look after, and and the right temperature and all of that. Unless you can do that, then you'll get worse than the local. So I really, and this is what the people in the Ilri are saying, is to work with local, a bit of crossbreeding, but otherwise work with your local genetic resources and start to do genetic improvement in those. That the Holstein Friesians are not necessarily your salvation. But again, that is a bit of a personal view, but part of that's from seeing it. As I said, I was in Tibet a number of years ago, and I saw all of these Holstein Friesians have been brought in, and they were doing far worse than the local breed of cattle because the farmers just could not maintain them. So that's, but that's a personal perspective.
Yeah, but in our country, there is a problem in our local breed because they are not suitable for commercial farming. They only give milk from 700 to 1,000 liters per lactation. Then what to do with?
Well, maybe they could be genetically improved. I don't know.
But genetic improvement of our local breed needs actually time, also almost 20-30 years. But in the meantime, how our farmer will survive?
These are long-term solutions. I agree. It's not a quick fix. It's not a quick fix. Yeah.
No, but what about crossbreeding, and how it can be solved by crossbreeding?
Yeah, this is the only way, Malik Umar Bhai.
Yeah.
I'll take you on that. Yeah. Okay.
Thank you.
Yeah, that's fine.
Thank you all. From now, Dr. Mijan will continue the event, and I will say goodbye to everybody. Thank you very much, everyone, for having here, and goodbye, Peter. Assalamu alaikum. Stay well. Bye.
Thank you, Mona.
Assalamu alaikum. Goodbye to all. Thank you.
Okay. Thank you.
Yes.
So, do I stay on or?
Sorry, Peter. We have a problem here. Yeah, we have a problem with the internet connection.
Right.
So, yes, and we had to stop the other laptop. So I couldn't follow the program anyway, but I can follow it later on the YouTube, no problem.
Sure. No, that's fine.
So
Okay.
yes, so I hope everything went fine.
Yeah, no, that was good. So, as I said, it was a personal perspective. So I hope it, maybe offered some insight. And look, thank you everyone for listening and your questions.
Yes, and if anybody wants to talk with you now, maybe we can. We are going to stop the recording very soon. And if anybody wants to talk here, they can talk. They can stay and talk with you. So just please stay here.
Sure, no, that's fine.
And we are going to stop the recording.
Okay.
So you can continue on formal talking. So it will be okay. So can I request Delwa to stop the recording? Delwa?
Okay, sir.
Okay, please stop the recording.